What are noncoding RNAs? Most genes code for a protein. The DNA is transcribed into messenger RNA, and the mRNA is then translated into a protein. For some genes, the final product is an RNA rather than a protein. These are noncoding RNAs. The DNA is transcribed into an RNA, and then that RNA could be processed or folded into a particular shape to perform its function in the cell. Some examples of noncoding RNAs are ribosomal RNAs, or rRNAs, and transfer RNAs, tRNAs, which are involved in translation. Small nuclear RNAs and small nucleolar RNAs are involved in RNA processing and modification, and long non-coding RNAs, microRNAs, miRNAs, and siRNAs, short interfering RNAs, are some examples of RNAs involved in gene regulation. Now we'll look more closely at the mechanism of gene regulation by miRNAs and siRNAs. miRNAs are encoded in the genome and are transcribed by RNA polymerase II, the same polymerase that transcribes mRNAs. The RNA is processed and exported into the cytoplasm. siRNAs are derived from viruses and are not encoded in eukaryotic genomes. They can be synthesized by scientists and placed into the cytoplasm of cells using various biotechnology methods. Both miRNAs and siRNAs are cleaved by dicer and then associate with the RNA RISC complex of proteins. The RISC RNA complex associates with its target mRNA, and the mRNA is either cleaved and degraded, or translation of the mRNA is inhibited. siRNAs are usually fully complementary to their target mRNA. When siRNAs, along with the RISC complex and the associated proteins, bind to the target mRNA is cleaved and then degraded. This degraded mRNA cannot be translated, so siRNAs prevent the protein product from being made. MicroRNAs are usually partially complementary to their targets. This partial complementarity allows them to target multiple mRNAs. When microRNAs, along with the RISC complex and associated proteins, bind to the target mRNA, translation is inhibited on the target mRNA, or sometimes the microRNAs target cleavage and degradation of the target mRNA. Both inhibiting translation and degrading the mRNA prevent or decrease the amount of protein product that is made.